Thank you for coming to see the FASEB webinar on Shared Resources 101. I will be starting the webinar in just a few minutes. Thanks. Good afternoon and welcome to FASEB's webinar on Shared Resources 101. Today, we'll be discussing ways to locate different types of resources, what programs exist to support instrument acquisition, and what you can do to promote sustainable sharing. All the FASEB resources featured today can be accessed on the FASEB website at faseb.org/sharedresources. I'm Bethany Drehman, the Manager of Data Analytics at FASEB, and I'm joined today by Sheenah Mische, the Senior Director of Advanced Research Technologies, and also the Associate Professor in the Department of Pathology at NYU Langone Health. We will have a Q&A session at the end of this webinar, but you can submit a question at any time as we go. To do so, simply go to the GoToWebinar panel. To view the whole panel, you may have to click on the orange arrow. Then go to the tab labeled Questions. Make sure that's expanded. Type your question in the box and hit Submit, and we'll be able to see it. FASEB, or the Federation of American Societies for Experimental Biology, is a federation of 30 scientific societies. We represent over 130,000 researchers, and our mission is to advance health and welfare by promoting progress and education in biological and biomedical sciences through service to our member societies and collaborative advocacy. Here are all FASEB societies, and their support and collaboration are essential to all of FASEB's work. If you're not already a member of a FASEB society, please check one of them out after this webinar. And now I'd like to hand this off to my co-host, Sheenah Mische.
Hello, everyone. So what are shared research resources? Across universities and academic medical centers, expensive research instruments and equipment are organized as shared research resources to provide access to technologies, equipment, and expert consultation, often on a fee or reimbursement basis, to enable, facilitate, and enhance the research mission of the university. Accessibility is a critical aspect of the definition of shared research resources. The scientific importance of such resources must not be underestimated. Scientific research resources, are critical to the research mission of our institutions, and new core facilities and shared sources have been shown to be the most efficient and economically feasible method for introducing new technology and expertise to drive research and technology advances. So why are shared resources important? Last fall, FASEB issued a report on maximizing shared research resources. As part of this report, we surveyed resource users and providers and determined that resource sharing provides many benefits to the research enterprise, particularly cost savings and expanded access to advanced technologies. The value and importance of optimal management in achieving highest efficiency, two aspects that strengthen biomedical research and the pace of scientific discoveries, are not always appreciated. Scientific research resources avoid duplication of shared resource services and identify and implement approaches that enhance their efficiencies. Given the funding agency budget constraints, it's important to determine how to support the best science with the available resources in partnership. We also found that shared resource facilities can promote quality control and scientific rigor, conduct technology development, serve as a nexus for collaboration and team science, and educate and train researchers. We recognize that shared resource facilities create a level playing field for all investigators by reducing the technical knowledge and cost barriers to implementing appropriate scientific approaches to their research. Where appropriate, investigators who acquire new instrumentation are encouraged to consider housing that instrument within an existing shared research resource to ensure maximization of usage for the highest impact on research and greatest benefits for training, sustainability of operation, and a high level of technical support. Bethany?
The report and many other materials we'll be discussing today can again be found on the FASEB website at faseb.org/shared-resources. So first, we'll jump in with locating resources. Our reports show that many researchers have trouble locating what resources they need. So we'll go over a few strategies to find the resource you're looking for. And I encourage you after this webinar to take a few minutes and explore some type of resource you haven't used before, because we'll be showing you a lot of different things that you might not have come across yet. Sheenah?
Okay, thank you, Bethany. When most people think of shared resources, they think of core facilities. But how do you find them? We found that many researchers rely on word of mouth, but if your colleagues don't have any suggestions, there are other places to look. First, try searching the ABRF core marketplace. The Vermont Genetics Network, in partnership with ABRF, established the core marketplace database to track and explore research services and technologies that are regionally accessible. And the ABRF core marketplace is now an international listing of 713 cores and used to view core laboratory capabilities, access core websites, and email telephone communication with the cores. And that link is provided here in this presentation. Next, check out your institution's list of cores. Many research institutions maintain this type of list, but they vary in quality. In the wake of FASEB's Maximizing Shared Resources report, many institutions have evaluated their core facility portfolios, are developing inventories of core activities, and indexing their services within and across organizational units. If your institution's list is difficult to find or use, advocate for its improvement and point to our report that describes how researchers are more likely to refer to user-friendly sites. You'll be helping not only yourself, but also the core facilities and other investigators. Finally, the Eagle Eye Network was developed in 2010 by a consortium of 9 universities with a grant from NIH. It represents over 40 research institutions, and continued technical and infrastructure support has been funded by the Harvard Catalyst at Harvard Medical School. Some facilities may be listed in the ABRF database, others in Eagle Eye or an institutional database. Still others may be in none above. So we encourage you to use all of these resources when searching for a core facility.
Another type of shared resource is stock centers and living collections. These provide research organisms to scientists around the country. To fill a void, FASEB developed its database of U.S. providers of research organisms, which can be found at the link on this slide. Other places, and this database includes more than 130 entries of providers, including academic and commercial providers, as well as all domains and kingdoms of life. You can email us at any time. The email is included on the database page to add or update a listing. We are always welcoming edits because we want to make sure this is an up-to-date resource at all times. If you're looking for a collection overseas, I'd suggest starting with the WFCC Global Catalog of Microorganisms. It is limited to microorganisms, but it does include many international listings that are not part of FASEB's US database. Another great resource is the United States Culture Collection Network. And Sheenah, I'd like to hand it to you to discuss national laboratories.
Okay, great. So national laboratories. User facilities of the national laboratories function as our nation's cores. A user facility is a federally sponsored research facility, and it's available for external use to advance scientific or technical knowledge. It's open to all interested potential users without regard to nationality or institutional affiliation. The allocation of the resources is determined by merit review of the proposed work, and user fees are not charged for non-proprietary work if the user intends to publish the research results in the open literature. Full cost recovery is required for proprietary work. So the Department of Energy has 3 biological and environmental research user facilities, as well as several synchrotrons at other facilities, which are used by structural biologists. Visit the user facilities webpage to learn more about the different technologies available, as shown here. And then the National Science Foundation, or NSF, also supports several national facilities, including the new National Ecological Observatory Network. There are no user-friendly portals for the NSF facilities, but most have their own websites. Now, the natural history specimen collections, the FASEB database covers only our living collections, but if you're looking for natural history or specimen collections, we suggest you start your search with these 4 databases listed here. iDigBio is an NSF-sponsored database of digitized records. Not only can you see where and when a specimen was collected and where it is located, you can also download a high-resolution image of the specimen. Other places to look for natural history collections include the Global Diversity Information Facility, or gbif.org, the AvertNet, and the Smithsonian Museum of Natural History Search Tool. Bethany?
For databases, unfortunately, there's no one-stop shop that we can point you to locate a database or data set. But progress is being made in this area. As part of the NIH Big Data to Knowledge Initiative, or BD2K, NIH funded the development of a data discovery index. The beta version of this tool is called DataMed, and is intended to become the PubMed for data. You can enter a protein name, disease, topic, or much more into the search bar. The results are all in the index datasets related to your query terms. DataMed indexes entries from 75 databases and repositories, including familiar ones such as Formbase and dbGaP, as well as ones created through the NIH Common Fund projects or other single program databases. The BD2K program has also sponsored a number of data-related tools, which can be found on the resource page. And if you're still searching for a database, I suggest looking at an article titled Biomedical Databases for Human Research. They have some very comprehensive tables of databases in that article. And of course, there's still more types of shared resources out there. The NIH Biomedical Technology Resource Facilities conduct research and development of new or improved technologies driven by the needs of all types of biological researchers. These centers are charged with making the technologies available to the research community in a sustainable manner, providing user training, and disseminating technologies and experimental results. You can see what centers and technologies are currently supported through the BTR portal. Another place to look for shared resources is the Resource Identification Portal, which was created as part of an initiative to generate persistent, unique identifiers for all research resources, thus promoting research identification, discovery, and reuse. It indexes model organisms, cell lines, antibodies, databases, software tools, and more. If your resources already have a unique ID, many journals, including the FASEB journal, support these IDs and publish manuscripts. The search function is a little finicky, so we suggest trying multiple queries. And some of these entries are derived from web crawling, so please independently confirm any results you find in it. And Sheenah?
Okay, thanks, Bethany. So, acquiring shared instruments. This is an updated FASEB report on the federal instrumentation grants and programs. The website is shown here. The FASEB's report, Maximizing Shared Research Resources, also documented the challenges with obtaining new instrumentation, as well as maintaining existing instruments. So in response, the FASEB Shared Research Resources Subcommittee updated this report on federal instrumentation grants and programs. It too can be downloaded from the FASEB Shared Resource page. Please note that the federal instrumentation funding is limited and that this document is only meant to help people locate relevant programs and does not guarantee success for your grant application. On the next slide, we show this is the current version of the instrumentation grant report. Please make sure the file that you're using is dated April 25th, 2018, as shown here. Bethany?
Thank you, Sheenah. The first section lists major grant programs for instrumentation acquisition, and they're organized by the funding agency. The table details eligibility, cost sharing, and other requirements, what amount of equipment costs are supported, and provides hyperlinks to the current program webpage, as well as to lists of past awards, and much more. We encourage potential applicants to review what these programs have previously funded when preparing your proposal. Since we first issued this report 2 years ago, NIH has started a new program for shared instrumentation for animal research, or CFAR. NSF has also changed the tracks for its Major Research Instrumentation Program, or MRI. The tracks now correspond to the requested funding amount, rather than if the application is for acquisition or development. So please be sure to double check the announcements before you submit your proposal to make sure it's the new layout.
Okay, thank you. So upon request, we added a new section on suggested best practices for developing a shared instrumentation proposal. In our experience, stronger proposals describe or include the following: a justification of need, broader impacts, instrument selection, technical expertise and human development opportunities, potential for methodology or technology development, sources of support for maintenance and repair, and letters of support from high-level institutional officials. We encourage you to share this page with co-applicants and relevant institutional officials as you prepare your proposal. And under instrument development, Federal research agencies also support the development of new technologies, instruments, and methodologies through specialized grant programs. Since we first issued this report, the NIH National Institute for General Medical Sciences, NIGMS, has added 2 new funding opportunities. NSF has been revamping some of its research infrastructure grant programs, but a new solicitation for their instrumentation development for biological research, the IDBR program, should be released later this year. Bethany?
Thank you. The remaining sections of this report detail instrumentation award programs for underrepresented institutions, secondhand instrumentation programs, but your institution may have a parallel internal program as well, and also a section on ways to locate user and core facilities. This last section includes links to many of the resources and databases we discussed earlier. Each year, funding agencies invest substantial resources in these core and user facilities to provide access to cutting-edge technologies, expert consultation, and other services for scientific investigators. Those services can include experimental design and evaluation needs, biostatistics, patient outreach, clinical regulatory issues, and many other types of support you might not necessarily expect to come from a core. And this FASEB document we just shared with you only covers programs for instrumentation grants. But as I just mentioned, there is also a lot of support for core facilities, living collections, and much more. For example, if you're a facility director, you're probably quite familiar with the NIH P30 mechanism, which is a major source of support for cores. Today, though, I'd like to call attention to a few of the lesser-known grant programs and encourage you to seek out some of the other niche opportunities that are out there. The National Cancer Institute recently started a grant program for staff scientists and core facilities conducting cancer research. There is a parallel program for laboratory-based scientists. NSF also has a specialized postdoctoral research fellowship that supports postdocs pursuing research in one of 3 specific areas, including research using biological collections. Outside of funding personnel, there are also programs to fulfill specific resource, acquisition, and development goals. For example, NIH recently sponsored the creation of 3 new cryo-EM service centers through an NIH Common Fund program, and also established a grant program for developing assays for small molecule detection. Sheenah?
However, the resource funding is limited. Resource sharing grew out of the need for access to technology and expertise. Consolidating and centralizing mandate from NIH was to drive efficiencies for collaboration to tackle big problems. But our biggest challenge remains in terms of funding, that the funding is available for the shared instrumentation, but it is not available for salaries, for expertise, or for long-term maintenance of those instruments. So with limited resource funding, it's critical that both the funding agencies and the research institutions receiving support from these resources identify and implement approaches that enhance the core resource efficiencies. The FASEB report, Maximizing Shared Research Resources, highlights recommendations from respondents, including encouraging grant applicants to propose the use of shared resources instead of instrument requests for their individual labs, allowing service contracts and a portion of staff salaries to be charged to equipment grants, and overall increased funding for shared instrumentation grants, including funding for less expensive equipment or sets of equipment that don't meet the system standard. And also, in terms of the major user group, it would be great for the NIH S10 applicants to document the full spectrum, demonstrating the breadth and stability of their NIH-supported user base instead of just focused on a major user group. So we need to enhance support because efficient use of shared resource dollars benefits the whole scientific enterprise. First, We support establishing a joint national strategy across all federal research sponsors, such as NIH, NSF, DOE, and the USDA. Many shared resources are used by multiple fields of research, so coordination would lead to more efficient use of limited funds. Institutions should also align their internal efforts for shared resource grant applications. This means that there is a group, ideally comprised of researchers, core facility personnel, and institutional leadership, that reviews proposals before they are submitted and considers to what extent the institution can support and utilize the requested resource. This approach facilitates transparency, helps institutions and sponsors avoid duplication, and encourages long-term planning for these capital expenditures. It could also help strengthen applications, as this group could identify additional potential users and may even be able to establish an institutional commitment for physical space, maintenance funding, and other necessary infrastructure. This approach facilitates transparency and helps clearly establish institutional need. We also need to promote interinstitutional sharing and regional core facilities. While not all core facilities can effectively serve investigators outside of their home institution, many can, and some institutions already have established MOUs to facilitate use at each other's facilities. We need to encourage more efforts like that, which will likely require creating incentives for institutions and resource users. And finally, researchers and institutions need to implement best practices for shared resources, from instrumentation, grant reporting, to core evaluations and strategic planning. Through good management and thoughtful planning, we can maximize the value of existing resources and better inform future expenditures. On the next slide, including for core scientists, it's one of our biggest challenges for support of shared resources is to establish support for the core scientists. These individuals make intellectual contributions to a broad array of research projects and lend their technical expertise to many more. However, there are very few sources for funding to support these scientists beyond fee-for-service, limiting their ability to conduct research and technology development. They also receive little professional recognition for their contributions and experience a high degree of career instability. It also affects retainment in terms of this highly skilled group of core scientists. By professionalizing this unique career track, we can promote excellence and make better use of these talented individuals. Research sponsors can implement grant programs like the R50 award mentioned previously that support research by core-based scientists. Sponsors can also offer training and development grants that would allow these scientists to gain new technical skills and keep up in their research field. Several institutions have established a professional track with a clear advancement pathway for core scientists, and we encourage other institutions to follow their lead. By doing so, institutions can retain and grow their core scientist talent. Bethany?
Thank you, Sheenah. As Sheenah described, funding and support for shared resources is limited. So it is incredibly important that we make sure our resources are sustainable. Today we'll be going over a few tips that researchers can do to help promote sustainable sharing. But afterwards, we also encourage you to check out FASEB's report for additional ideas. And Sheenah?
Okay, thanks, Bethany. So, ensuring acknowledgement. We know that acknowledgement metrics are used for assessment of cores and shared research resources. They're also used for career advancement decisions. Core directors and staff are scientists and experts in their field. When they make a substantial intellectual or an experimental contribution to a publication, they deserve to be acknowledged or included as a co-author. Institutions and sponsors measure acknowledgements and use them to inform decisions affecting funding and career advancement. Thus, through proper acknowledgement, investigators play a critical role in ensuring their continued availability and future development. Acknowledging expertise and data generated in a core contributes to the rigorous and transparent reporting of research methodologies and results. Examples of these core-based practices include unbiased acquisition of analysis of data, standardization of instrumentation and methods, best practices for data management and curation, and the QA/QC of key biologicals and chemical resources. On the next slide, Bethany will talk about how FASEB can help.
Yes, we know that researchers agree that providing appropriate acknowledgement for core facilities and the core scientists to help with the project is important, but acknowledgement can often be overlooked when you're in the rush to assemble all the different parts of a manuscript or grant application. So we developed this one-page tool to help you find ways to ensure that that acknowledgement doesn't slip through the cracks. It includes ideas for researchers, core personnel, institutions, and publishers. For example, we suggest that facilities provide standardized acknowledgement text on their facility site, reports, bills, and other communications so that researchers can easily find it, copy and paste it into their document, and be done. We also have several recommendations for researchers, including just taking the simple step of checking in with all the co-authors what facilities they did use to collect their data before you finalize your manuscript. And Sheenah will talk about authorship.
So ABRF, the Association of Biomolecular Resource Facilities, provides recommended guidelines for authorship on manuscripts. This can be found on their website, which is shown here under Authorship and Guidelines. The Authorship Guidelines provides recommended guidelines for authorship, and this was published in 1999 by Ruth Angeletti et al. Essentially, intellectual interactions between resource and research scientists are essential to the success of each project. When this success results in publication, a citation in the acknowledgement section of a manuscript may be appropriate for routine analysis. However, contributions from resource scientists that involve novel resource laboratory work and insight, experimental design, or advanced data analysis that makes a publication possible or significantly enhance its value requires co-authorship as an appropriate acknowledgement. The ABRF webpage covers scientific contributions that constitute authorship versus acknowledgement, some useful examples, and some useful practices for cores as and shared resources, and a compendium of supplementary information and resources. Bethany?
Thank you, Sheenah. Another way to promote sustainability of core resources is to increase the visibility of the resource and the core scientists themselves. And there are many ways to bring them into various institutional activities you're already involved with. For example, in recruitment, having core scientists serve on the faculty recruitment committee helps involve them in decision-making processes, but we also know that cores can be a big selling point, and so it's great to have them in the room so they can share what is really available. Of course, their technical expertise makes them a great guest lecturer and can increase awareness of the shared resource among students. Similarly, include them in the planning and holding of orientations for graduate students, postdocs, staff scientists, and faculty, and even include them on the strategic planning committees, whether departmental or institutional, since these resources serve institutional researchers. And this should go without saying, but we didn't want to leave it off. Also include them in your research. Discuss your research aims and resource needs with staff and core scientists. They might have ideas, suggestions, or even techniques that you have not even thought of, and they can be a valuable contributor to your research project. And finally, Sheenah will be talking about how to create change through your mentoring and leadership.
Thanks, Bethany. So we know that our core scientists are multidisciplinary experts. And we know that multidisciplinary scientific collaboration is the cornerstone to effectively address human health questions. And it really is the hallmark of biomedical research. So accessing that expertise and shared resources is critical to accelerate discoveries in scientific research. Cores support transparent processes for knowledge and data management. They are integral to faculty recruitment and retention. They level the playing field for those young investigators, and they are integral to the education and training of our next generation of scientists. So researchers can also help transform how the next generation views shared resources and the professionals who provide them. It is a partnership, and by developing collaboration and team science with our core scientists, you are leading by example, and this is becoming more and more commonplace. Likewise, support authorship policies that include shared resource scientists based on their contributions. Next, serve on mentoring committees for the faculty core scientists. And finally, teach trainees how to find shared resources, including institutional and national resources. This will help prepare trainees to become independent researchers. And as a first step, you could share this webinar and the resources that we featured here. The FASEB report and recommendations, there are many strategies to make shared resources more sustainable, and we can't cover them all in this 1-hour webinar. For more ideas, please refer to the FASEB report, Maximizing Shared Research Resources. We identified and offer recommendations in 4 major areas: funding and business operations, discoverability and access, ability to meet evolving needs, and facility, career track, and staff development. Part 1 presents our 10 recommendations to optimize sharing, and Part 2 contains our discussion and survey analysis. We thank all of the scientists and people in FASEB societies who contributed to this effort. And if your institution or department is looking to optimize resource sharing, please send them a copy of this report. Bethany?
Thank you again. Yeah. Thank you again for joining FASEB's webinar today. We'll be now moving to the questions and answer portion. I'll be reading the questions, and then Sheenah and I will be jointly answering them. So our first question is, will the slides be made available? Yes, we will be posting the recording within the next week and making the slides available online. Please allow us a few days to get that all up and sent out, but you should be receiving an email from us shortly with that information. The next question is, my institution does not value or support shared resources. What can I do? Well, I would start with just documenting and demonstrating the value of shared resources. You can track the number of users, number of uses, publications that acknowledge the shared resource, grant applications that cite the resource in the grant application text. But part of that involves you acknowledging those resources too ahead of time, so that record is there. You can also document cost efficiencies and also share the FASEB report about maximizing shared resources. Sheenah, what other ideas do you have?
Bethany, I think there are a number of excellent recommendations that came out of this survey and the report. One thing that really was highlighted in the report was the fact that the core scientists really are their best advocates, and also in terms of getting your customer base to basically share what they have, how they have benefited from using the core. There were also recommendations in terms of getting out, presenting, what the core capabilities and expertise is, at department meetings. Institutions have done a really good job in terms of promoting a core day or a series of seminars that would highlight some of the research that has been generated in the cores to get the information out there, because we know that the hardest part is really identifying what are those resources on campus, and sometimes it's just a matter of getting out there and marketing what your core does and being your best advertiser, so to speak.
Thank you, Sheenah. We have another question. This is, our cores are staffed by faculty scientists. They have the added responsibility of seeking their own grant funding and sustaining their own research. What would be some of the best approaches to sustaining their own research while effectively supporting core needs, including research and development? Well, as we discussed today, there is limited support for research by core scientists, but there are a few. So I would definitely encourage pursuing some of those. But also, some institutions do provide core scientists with some protected time outside of fee-for-use, reimbursable work. So you might look into whether your institution has that for other cores and whether that could be extended to your core if they would consider building that in. Sheenah, what are your thoughts?
So again, what we learned from the survey is that if there are 100 institutions, there are 100 different ways to be able to look at how a core is supported financially. And part of this mix has to do with being a faculty member, having responsibility in terms of finding funding to support your research that would ultimately benefit your career as well as the advancement of the development of the core. And so the most successful cores that have this approach is, as Bethany talked about, which is a protected time, whether it be 30% or 40%, and that the remainder of it is devoted to the development of services that cost can be recovered for with a fee-for-service model or a cost recovery model, which of course is compliant with the regulations from the Office of Management and Budget and the Unified Guidelines. And so as any core director who is a faculty member or just a core director knows, it is a balancing act between providing cutting-edge research and science support, as well as being able to continue to develop, what is happening in the field, as well as to continue to develop your career. And so again, we go back to making sure that those acknowledgements happen, making sure that you effectively are considered or included as co-authors, that you are publishing your own research out of your own funded research, and that you continually push the envelope as far as wearing the many hats that a core director wears, never mind, the business side and the marketing side.
Our next question is, the R50 award looks like a good opportunity for core staff, but what about support for core directors? It looks like they already have to be a PI to be eligible. So I would note that just because you're a core director doesn't mean you can't be a PI on a research grant or a co-PI on a research grant. You don't have to stick to just grants that are specifically targeting cores. Maybe you want to look into some of the instrumentation development grants we talked about. Some of them also support just methodology development. So there are grant opportunities outside of your standard core-specific opportunities that might be able to support core directors as they work on their own research projects and their own development projects. Sheenah?
And on that topic, I would encourage core directors to be an active participant in writing grants where you can be a co-investigator or you have a percent effort of your time on a particular grant, because it will expand your horizons, but it also strengthens the research at the institution, where you have core directors that are co-investigators on a number of different grants, as well as considering, specifically about the R50, there are some restrictions in terms of that funding, but whether it's for a staff scientist or whether it's for a core director, I would encourage you to read the details in terms of that RFA.
Okay, our next question is, what should I do if my core does not produce good results? Well, we've heard this story from both sides of the aisle, so to speak, from both core scientists as well as facility users. And as a first step, I would start by checking if it's a sample issue. Are you using the same protocols as the core in your sample preparation? We've heard all sorts of stories from things being over-diluted or not buffered properly or not neutralized, and that was affecting the results. So step one is to just work with the facility to try to solve the problem, including going over all the protocols. And as we discussed today, there are a lot of ways to find more core facilities. So if the one that's at your institution isn't meeting your need, there may be another one out there that can. So first, try to work it out with the core, but also be open to trying other facilities. Sheenah, do you have any other suggestions based on your extensive core experiences?
So what I would suggest is to work closely with the core. If this question was coming from an investigator who was, finding challenge working with their particular core, as Bethany said, it very often comes down to the experimental design. And so the time that you spend early on in that consultation about what are your scientific aims for this particular project, it helps enormously in terms of the success. Because remember, the core directors and the core staff have seen a wide range of samples, applications, methodology, and they really are the best source for knowledge management for the institution. But if you find that this is not a productive relationship with the core that you're working with, there are a plethora of resources that we mentioned today, including the ABRF Core Marketplace, Eagle Eye, and also just within your institution, because we did learn that the majority of people who learned about cores learned it from their very own colleagues and researchers at their institution, or perhaps at neighboring institutions, or where you have collaborations at other institutions.
And that is such a great point, Sheenah, about making sure you get it right from the start. It's better to talk with them before you start your project than be troubleshooting afterwards.
It's also a lot less expensive.
Yeah. We have another question as a follow-up to that. They suggest insist on the core providing system controls. It's often the samples, but the core needs to be able to show that their systems are working. I definitely agree with that. Many cores do provide that, but if yours doesn't, I think that is a great suggestion on what, as a first step in your troubleshooting, to look get those system controls as well as compare your protocols.
Absolutely. I could not agree more that cores are a very well-documented repository for best practices for scientific rigor and reproducibility. And so most cores would absolutely be responsible for that QA, QC, including standards, standardization of methods, documented workflow, as well as the QA, QC for those controls, for key biologicals, for the chemical resources. We also know that in very instrument-heavy core facility shared resource, they have high standards for the standardization of the instrumentation, which includes regular and rigorous QA and QC, and a very strict performance maintenance schedule that are on these instruments, because they have to be running 24/7. So it may not be obvious to you, but this is one of the discussion points early on in terms of what you are looking at. Very often this early discussion is all about setting expectations. You have expectations as a investigator, and the core director, core staff have expectations, and it's really about the communication to establish what will be the deliverables in terms of that very important data package. Bethany?
I think you covered it fully, but I would just add, if you do check the FASEB report, Maximizing Shared Research Resources, one of the things we did find through our survey was that core personnel were much more likely than people in the lab to report that they were confident in knowing what resources and instruments needed to be validated, calibrated, etc., and were much more likely to report that their own unit had standardized protocols for validation, calibration, etc., for all the resources and all the instruments they use. So a lot of that QA, QC is built in at a core. I don't see any further questions, so we'll wait 1 minute, and if no more come in, we'll be wrapping up. So please, if you have a remaining question, get it in real quick. Well, thank you for joining us today on our webinar on shared research resources, and thank you, Sheenah, so much for co-hosting it with us.
And thank you, Bethany. I think that we shared a lot of really good information about a variety of resources that are out there.
We will be posting the slides and recorded webinar over the next week. And once those are live online, we will be sending an email to all the webinar participants today so that you can access them. Thank you again, and have a great afternoon.
Bye-bye.